Qubits, the fundamental building blocks of quantum technologies, have traditionally been associated with complex systems like diamond nanocrystals or quantum dots. However, a groundbreaking study introduces a new player in the field, fluorescent proteins as optically addressable spin qubits. This innovative approach leverages the metastable triplet state of enhanced yellow fluorescent protein, EYFP, to create a genetically encodable qubit sensor, opening up exciting possibilities for quantum sensing in biological systems. The challenge of biological quantum sensing. While quantum sensing has revolutionized physical sciences, its application in life sciences has been limited. Current platforms, such as nitrogen vacancy centers in diamond nanocrystals, offer exceptional sensitivity, but face challenges in biological measurements due to size, complex surface chemistry, and incompatibility with genetic techniques. The study highlights the need for a molecule-scale, genetically encodable qubit sensor to overcome these limitations. Fluorescent proteins as spin qubits. The researchers introduce fluorescent proteins, specifically EYFP, as a novel solution. These proteins, with a diameter of 3 nanometers, can be genetically encoded in various target systems. By fusing the fluorescent protein with a target protein, a deterministic tagging is achieved, allowing for precise correlation between the probe and biological target. This approach has already had a profound impact on life sciences, enabling unparalleled correlation compared to synthetic molecular fluorophores or nanoparticles. Optical addressability and spin readout. The study demonstrates the optical addressability of the EYFP spin state using a custom confocal microscope. By initializing the spin with a 488 nanometer optical pulse and employing a photophysical technique to shorten the triplet lifetime, the researchers enable on-demand spin readout. This optically activated delayed fluorescence (OADF) scheme significantly enhances readout speed and sensitivity. Magnetic resonance and coherence. Through optically detected magnetic resonance (ODMR) spectroscopy, the researchers investigate the metastable triplet spin state. They observe coherent Rabi oscillations and measure spin coherence times, showcasing the potential of EYFP as a quantum sensor. The study also explores the impact of magnetic fields on coherence and demonstrates the effectiveness of dynamical decoupling sequences in improving coherence. Room temperature operation and biological applications. The researchers successfully demonstrate room temperature operation of the EYFP qubit, highlighting its potential for biological applications. They express the qubit in human embryonic kidney cells and E. coli, performing spin measurements and observing high contrast in ODMR spectra. This genetically encodable qubit opens doors for probing biological processes, such as oxidation states of metalloproteins and drug-binding mechanisms. Future prospects and improvements. The study discusses potential enhancements in sensitivity through improved coherence, spin readout, and initialization. By optimizing optical readout and utilizing fluorescent cycling, significant improvements in signal-to-noise ratio are anticipated. Furthermore, the researchers speculate on the possibility of multiplexed imaging using the narrow, room temperature ODMR resonances of the fluorescent protein qubit, potentially enabling up to 100 orthogonal colors. Conclusion. This research establishes fluorescent proteins as a genetically encodable spin qubit platform, bridging the gap between quantum information science and bioengineering. With further developments in sensitivity and ferrostability, these protein-based qubits could revolutionize quantum sensing in biological systems, offering transformative possibilities in both fields.